everyone, and welcome to the Biophotonics Discovery Podcast. Whether you're a seasoned researcher, a student just starting out, or you just love a good story about how light is used to solve big problems, this podcast is for you. We're making the discoveries in biophotonics accessible one episode at a time.
Lighting Up Hidden Microbes, a new frontier in vaginal health monitoring by the Locke Lab at Vanderbilt University. A groundbreaking study has shown that light, in the form of surface-enhanced Raman spectroscopy, or SIRS, can reveal the hidden microbial shifts in vaginal fluid, potentially revolutionizing how we assess vaginal health at the point of care. Using a laser-based technique that captures biochemical fingerprints, this method may enable earlier and more accurate detection of microbial imbalances that affect millions of women worldwide. The vaginal microbiome, home to various bacteria, plays a vital role in maintaining reproductive health. Its breakdown, a condition known as dysbiosis, is linked to complications like bacterial vaginosis, BV, sexually transmitted infections, and even preterm birth. One bacterium, Lactobacillus iners, is commonly present in healthy vaginal ecosystems, but often goes undetected using standard diagnostic tools. Another, Gardnerella vaginalis, is overwhelmingly associated with BV. Current methods like gram staining or bacterial culture lack the sensitivity and specificity needed to detect these organisms reliably, especially in real-world clinical contexts. The Locke Lab, led by PI Andrea Locke at Vanderbilt University, aimed to fill this diagnostic gap. They collected vaginal fluid from participants at routine gynecology appointments and analyzed these samples using both a high-end Raman microscope and a portable spectrometer. They then used quantitative PCR to confirm the presence of L. iners and G. vaginalis, correlating that data with the vibrational spectra from SIRS. What emerged were distinct spectral signatures tied to microbial presence. When G. vaginalis was present, spectra revealed spikes in protein and lipid content and a drop in organic acids. In contrast, L. iners was associated with increased organic acid signals and dips in proteins, amino acids, and sugars. Crucially, these patterns were visible even when bacterial counts were below infection thresholds and before any clinical symptoms appeared. Even more promising, the portable spectrometer produced nearly identical results to the lab-grade system, suggesting the real possibility of a compact, point-of-care tool for in-office or home-based screening. We're not just finding infections when they've already taken hold, said lead author Anna Rourke-Funderburg. We're detecting early shifts in the microbiome, weeks or potentially months before symptoms begin. The implications are profound. Early detection of microbial imbalance could allow for timely intervention, reducing the risk of disease progression and improving outcomes across reproductive health. By proving SIRS can pick up on these subtle biochemical changes, this study not only validates the technique's diagnostic power, but also opens the door for broader, more accessible microbial monitoring in women's health. Looking ahead, the research team plans to expand sample sizes and explore additional bacterial species using full microbiome sequencing. For now, this study marks a major step towards smart, non-invasive monitoring of the vaginal microbiome and a hopeful future where personalized microbial insights are just a light scan away. For details, see the original Gold Open Access article by A. Rourke-Funderburg et al., investigating microbiota and biochemical changes in vaginal fluid toward point-of-care microbial monitoring using surface-enhanced Raman spectroscopy in Biophotonics Discovery.
If you enjoyed this episode, please subscribe, leave us a review, and share it with your colleagues. Your support helps us shine a light on the latest in biophotonics research. Stay curious, and we'll see you next time.